One of the major themes in this course is diversity and unity. Basically, life is very diverse, life is different, and yet there is a very striking unity. We all share DNA as a universal genetic language, and we all have similar cells, which are our basic units of structure and function. How do we study cells? Well, we use microscopes. Zacharias Janssen is credited with inventing the first microscope, possibly with the help of his father, and while very simple compared to today's microscope, was a big step. Second was Robert Hooke. He was the first cytologist, or the first person to study cells, and he actually coined the term cells in his book Microscopy. Looking at cork, he saw the shapes of those cells looked kind of like a honeycomb or the rooms that a monk lived in, which are called cells. And so it became the term that we used. The cell wasn't really understood for many years, but he still is credited with describing them. The next major step was Antony van Leeuwenhoek, which was a Dutch tradesman and scientist, commonly known as the father of microbiology and considered to be the first microbiologist. He's best known for his work on the improvement of the microscope and for his contributions towards the establishment of microbiology. Using his handcrafted microscopes, he was the first to observe and describe single-celled organisms, which he originally referred to as animalcules. He had read Microscopy, which prompted him to do this work with microscopes. He was the first to record observations of muscle fibers, bacteria, spermatozoa, and blood flow in capillaries. He didn't actually author any books of his own, but he did publish several letters to the Royal Society in London, which eventually published them and is how we know about his work. Today we take many things in science for granted. Many experiments have been performed and much knowledge has been accumulated that people didn't always know. For centuries, people based their beliefs on their interpretations of what they saw going on in the world around them without testing their ideas to determine the validity of these theories. In other words, they didn't use the scientific method to arrive at answers to their questions. Rather, the conclusions were based on untested observation. Among these ideas for centuries, since at least the time of Aristotle, people believed that simple living organisms could come into being by spontaneous generation. The leaf that falls on the water turns into a fish. Grain left in the barn will turn into mice. So on. The study of the cell began with the discovery of cells by Robert Hooke, who reported them in Microscopy. But that's not where it stopped. Lawrence Oken believed that all living beings originate from or consist of cells. Jean-Baptiste Lamarck expressed similar ideas and said that no body can have life if its constituent parts are not cellular tissue or are not formed by cellular tissue. In 1831, Robert Brown reported the presence of a nucleus in the cell, but three major scientists contribute to what we call the cell theory. German zoologist Theodor Schwann in 1839, a German botanist in Schleiden in 1838, working independently, came out with a theory called the cell theory. They said that the cell consists of three basic parts: the nucleus, the fluid or cytoplasm, and an outer thin covering, or the plasma membrane. The cell wall, they said, was an additional structure present in only plant cells. So the cell theory is one of the most fundamental generalizations in biology and had a wide-ranging effects in all fields of biological science. But it was eventually extended by Rudolf Virchow in 1855, who was a German physician, and he hypothesized that new cells were formed only by the division of previously existing living cells, omnis cellulae e cellulae. Cells come from cells, which was a very different idea from the abiogenesis that was prevalent since Aristotle. So the three major components of the cell theory are laid out here, and you will be required to know them. So looking at microscopes, we find that there are many types of microscopes. The unaided eye cannot see cell organelles, it cannot see atoms, so we use microscopes to see these smaller things. There are three major groupings of microscopes, but really only the first two you have to worry about. Optical microscopes use light. Compound microscopes use regular light, and it's the ones you're going to actually use in this class the most often. You can view individual cells, even living ones, and it has a very high magnification. However, it has a low resolution. And don't worry, we'll explain what those words mean on the next section. But another type of light microscope is the dissecting microscope. So if I want to examine a whole organism, be it a cricket or a mouse or some tissues, doesn't matter. You have a flat bed for laying out that organism, and it's much easier to work with. But this still uses light, regular visible light. The confocal microscope is a little more advanced and very expensive, and we do not actually have access to one of these unless we go over to Coastal. But this microscope uses laser light. Because the wavelength is so much smaller, it's able to see a lot more detailed and fine images that we couldn't see. Now, beyond the confocal microscopes, and you want to get into some of the smaller things, we need electron microscopes. There's two major types of electron microscopes. The TEM, or the transmission electron microscope, gives you a two-dimensional view. You take thin slices of the specimen and beam electrons through it. Now, it's slices, which means you cannot use living things in a TEM, unlike in a light microscope. The electron beams will pass through this. It's going to have a very high magnification and a very high resolution. The scanning electron is a little different. The image is seen in 3D. It has a very high magnification and resolution, but you coat the specimen in gold and you bounce electrons off of it to give you an exterior view of the specimen. So for this example, we have a rabbit trachea. You can see the difference between a TEM and an SEM image here. It's the same structures, completely different. Scanning probe microscopy and beyond is where we're going with in the future. Scanning probe microscopes cover a lot of different related technologies, very fine scales down to the level of molecules and groups of atoms. Basically, you have a sharp tip that moves across the object's surface and gives you a very sensitive image. Say, for example, this type, which is even further advanced, is a quantum microscope, which allows us to see an individual hydrogen atom. Microscopes enhance our sense of sight. They allow us to look directly at things that are far too small to view with the naked eye. They do this by making things appear bigger, magnifying them, and at the same time increasing the amount of detail we can see, increasing our ability to distinguish between two objects or resolve them. For this reasons, they are one of the most widely used tools in science. Different kinds of microscopes can show us different amounts of detail. Electron microscopes have a far greater resolving power than light microscopes, so we can use them to see even more detail than it is visible under a light microscope. To understand the difference between magnifying and increasing the detail, use this example. This image has been enlarged 64 times on the right. You can't see any more detail because the image is now pixelated and blurred. The naked eye can tell things apart at about a tenth of a millimeter. Any closer than that and they seem to be the same object. Under a light microscope, you can see things that are less than a micrometer apart, a thousandth of a millimeter. Now, scientists have worked out why we can't see an unlimited amount of detail, because if an object is less than half the wavelength of the microscope's illumination source, it's not visible. So since light has a wavelength of about 400 nanometers in a light microscope, you can't see anything smaller than 200 nanometers. So maybe a large organelle, but that's about it. The wavelength of electrons is thousands of times shorter than visible light, so scientists predicted that electron microscopes would be able to resolve objects that are thousands of times smaller, and they were right. Now electron microscopes can detect objects that are approximately 1/20th of a nanometer, 10 to the negative ninth meters in size. And we're getting better and better at that all the time. With some of the current probe microscopes, you can even see chemical bonding happening. So this is a pentacene molecule imaged with an atomic force microscope, which is a type of probe microscope. And you can actually see the carbon rings that made it up. One of the last ways of studying cells is by studying their subcomponents, because each organelle is specialized for a separate job. So we do that by fractionating the cell. Basically, you turn it into homogenate, you blend it up, and you spin it at different speeds. The heaviest particles will settle first, the lightest particles will settle last. So for example, nuclei and cellular debris will be settled out first, whereas ribosomes, which are the smallest organelle, are going to settle out last. So you homogenate it and then you spin it using an ultracentrifuge or a microcentrifuge depending on your goal. Ultracentrifuges are really, really fast, but they're really big, and they go about 1,000,000 times the force of gravity. And they have to have a foot thick lead walls because every now and then one of them disintegrates while it's spinning, and when they blow, they really blow. It's a lot of energy, and you end up with titanium shrapnel. Microcentrifuges are going to be for smaller scales, studying proteins, genes, DNA, and we actually have one of those if you would like to take a look.